Hello, everybody, and welcome back. We are in Lab 12 today, and I'm going to shoot this in two separate videos. All I'm going to do is part one right now, an intro, and then part one, and then I'm going to go outside and try to film part two. I'm not exactly sure how that one's going to work, so I'm just going to do part one because that's easy for us. And let me go ahead and pull up my screen here. And what we're doing in lab number 12 is natural selection. So on this lab, there's really only two exercises that we are going to be playing with. We're going to be looking over. So natural selection. And with this, again, we're only going to cover part one right now. You're going to need your lab manual with you to fill this one out because I'm not going to give you data to fill out table 12-1 and 12-2. But we're going to run through the experiment together. And so I'm going to actually run through it with you. And you're going to be filling out the data tables 12-1 and 12-2 as we go. So you're going to need that with you in a couple minutes. So if you're not there yet, go ahead and pause the video, pull out your lab manual, get to page 173, because we're going to need that in just a few minutes. So the first part of the lab and the introduction to that lab talks about the theory of evolution by means of natural selection. And there were two people who were very influential in essentially developing the theory of evolution, at least by means of natural selection. The guy on the left, Charles Darwin, he generally gets most of the credit for this. He's the one who wrote his book on the origin of species by means of natural selection. But there was somebody else, this guy on the right named Alfred Russel Wallace, who was essentially working with different organisms, but came up with pretty much the identical theory as Darwin in terms of how species can possibly change or evolve over evolutionary time. Now, both men essentially, like I said, they came up with the same mechanism and how it works, and there's two conditions which would have to be met in order for natural selection or evolution to take place by means of natural selection. One condition is overproduction of offspring. And what that means is that within a generation, the parents produce more offspring within that generation than it takes to replace the parents. And that's actually pretty easy to show. Because imagine you had a population where all the individuals in that population paired up. And so equal sex ratio, everybody paired up. And if they produce just one offspring per couple, what would happen to the generation size, or the size of the population in the next generation? Yeah, it would cut in half. So you'd actually have the population decreasing. So all species, at least species which are either stable or increasing in their population size, produce more offspring than within a generation than it takes to replace the parents on average. And the interesting thing about that is that most wild populations are relatively stable over the large scale. So while there might be year-to-year fluctuation within population size, they generally, again, most species generally stay about the same. And so if we have this overproduction of offspring, producing more offspring than it takes to replace the parents, what that means is that not all of those offspring will actually be able to survive and reproduce themselves. Some will either die or just won't be able to reproduce as they get older. So that's the first condition, overproduction of offspring. The second condition that both Darwin and Alfred Russel Wallace argued for is that there's individual variation among the members within a species and within a population. And much of that variation is heritable, which means it's located on the chromosomes, it's a genetic component, and it can be passed down from one generation to the next. And what that individual variation does, it essentially creates the genetic variation within the population such that some individuals are likely to be better adapted to whatever the environmental conditions are at that time. And there's many, many, many factors coming into play. It's not just one character, one condition, but there's multiple different physiological and anatomical traits which can contribute to this. So individual variation, which is genetically derived, so it's heritable, can be passed down from one generation to the next, and overproduction of offspring, where not all individuals that are produced survive to reproduce themselves. And so if you look at that combination of those two, what you end up getting is something that they called unequal reproductive success. And unequal reproductive success simply means that some individuals are more likely to survive with and reproduce than others within a population or within a species. And they say that, or they propose that the individuals that have the heritable characteristics, so the genetic makeup which makes them better adapted, they're more likely to survive and reproduce than the individuals which have genetic components or physical traits, heritable characteristics that make them not as well adapted to those current environmental conditions, whatever they happen to be. So essentially, again, let me summarize: overproduction of offspring, producing more offspring than it takes to replace the parents. Individual variation among the offspring leads to unequal reproductive success, where some individuals are more likely to survive and reproduce than others. And that is one of several mechanisms by which evolution can occur. That's the basic premise behind how natural selection works. Which brings me to the question of, well, what is evolution? And if we're thinking about what evolution is, if we look at microevolution, what microevolution actually is, it's the change in the allele frequency within a population. So if you have two alleles for a gene, and the frequency of either the dominant allele or the recessive allele changes over time, well, that's one way that we can define evolution. If the allele frequency changes, then we're said that population is evolving. So how does the allele frequency change? There's a number of different ways, and in this lab, in Lab 12, we're going to look at two different examples of how the allele frequency can change. And one of those is by looking at deleterious alleles within a population. So deleterious, delete. So deleterious are alleles that are lethal. So an allele that is deadly is considered a deleterious allele. In this particular exercise, we're going to be applying essentially standard Mendelian genetics, where we have two alleles making up a gene. One allele is going to be dominant, one's going to be recessive. So we're dealing with complete dominance in this particular example. And with complete dominance, again, as a reminder, the homozygous dominant condition and the heterozygous condition, or the homozygous dominant and the heterozygous genotypes, are both expressed as the dominant phenotype. And then the homozygous recessive genotype is expressed as a recessive phenotype. So again, this is standard Mendelian genetics, the basic stuff that we talked about in lecture earlier this semester. All right, so for this first exercise, we're going to be looking at deleterious alleles as a source of evolution, and we're going to play with a population of marbles. If you've read over the instructions in the lab manual, basically what you're going to have is a population or a bucket full of marbles. There's supposed to be 100 marbles in the population, 50 black marbles, 50 white marbles. I counted mine. I had 48 black and 50 white, so I took 2 out. So now I have an equal number of marbles within my population. For both of these examples, for Table 12-1 and Table 12-2, we're going to say that the black marble is the dominant marble. That's a dominant allele, whereas the white marble represents the recessive allele. And in round 1, we're going to say that the recessive allele is deleterious, or the white marble is deleterious. So I'm going to head and close my screen here for just a second, pause that share, stop that, and go back to the video, and I'll show you what we're doing here. So here we are. Here's my population of marbles. I have 96 marbles in here, 48 black, 48 white, an equal number of marbles, and I've got 3 plastic cups. And what I'm going to do is essentially decide which one of these marbles survives and which one of these marbles die, or individuals. So in this population, we're letting the marbles represent individuals, and it takes 2 marbles to represent 1 individual. Again, we're looking at just 1 gene. We're looking at the 2 alleles for that gene. And so what I'm going to do is I'm going to pull out 2 marbles at a time. I'm going to show them to you, and I'm going to drop them into one of my 3 plastic cups that I see here. That you see here. And so the 3 possible combinations that I can do is I can pull out, I'm going to cheat here and look, I can pull out 2 black marbles. And those 2 black marbles, those are homozygous dominant. That's going to go into my first cup, my dominant cup. I can pull out a heterozygote condition, 1 black, 1 white. I would put that into my heterozygote cup. And again, I'm cheating. I'm going to pull out 2 white marbles here, and the 2 white marbles is homozygote recessive. What happens to this individual who's homozygous recessive? Die. Yeah, so it's going to go to my third plastic cup. That's going to go into my death cup. And what I'm going to do is I'm going to go through all of these. I'm actually going to dump my 6 marbles back in, my individuals back in. I'm going to go through all of these, and I'm just going to pull them out, hold them up to the screen. I'll say dominant, heterozygous, or recessive, and then I'm going to drop them into my 3 cups. As I do that for every marble pair, you should put a little tick mark on your table 12-1 for your first generation, and we'll see how many generations it takes to rid ourselves of the deleterious allele when it's the recessive allele. So all of my marbles are going back in. I'm going to mix it up. And this is what we're doing in lab. So this is what we're doing right here. So my first marble pair, dominant. Hetero. Hetero. Recessive. Hetero. Dominant. Recessive. Dominant. Recessive. Recessive. Hetero. Recessive. Dominant. Dominant. Hetero. Dominant. Recessive. Hetero. Hetero. Dominant. Hetero. 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 Recessive. Hetero. Recessive. Dominant. Recessive. Hetero. Recessive. Dominant. Hetero. Dominant. Dominant. Hetero. 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 Recessive. Recessive. Hetero. Recessive. Dominant. Recessive. Dominant. Dominant. Hetero. And my last pair is hetero. All right. So what I have, all black marbles in 1 cup, even mix black and white marbles, heterozygous in my 2nd cup, and all white marbles in my 3rd cup. So these guys have the dominant allele, and again, for this first condition, the deleterious allele is recessive. So these guys survive. Let me go back into my population. My heterozygote individuals, because they have at least 1 dominant allele, that dominant allele provides protection. They survive as well. They go back in. But my all-white marbles, they died. So I'm not going to put them back in the population because they're dead. We're going to keep those off on the side. I'm going to stir up my bucket. Stir those up, put my 3 cups back in order, and now we're at generation 2. Same thing. Hetero. 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 Dominant. Dominant. Hetero. Hetero. Dominant. Dominant. Hetero. Hetero. Dominant. Hetero. Hetero. Dominant. Recessive. Hetero. Hetero. Dominant. Dominant. dominant, hetero, 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 dominant, hetero, recessive, dominant, 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 hetero, and dominant. All right, so we saw that one, the population was smaller, so instead of having the originally 48, 49 individuals, we cut that down, and we had fewer individuals who were homozygote recessive, so fewer individuals died that time. I'm going to do the same thing, put all my surviving black marbles back in, put all my heterozygotes back in, I'm going to mix it up, my white marbles stay out, they're still dead, we're going to go ahead and run through generation 3. All right, start generation 3, table 12-1, and dominant, 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 hetero, dominant, hetero, dominant, hetero, dominant, hetero, dominant, 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 recessive, hetero, dominant, 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 hetero, hetero, dominant, hetero, 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 dominant, recessive, hetero, and dominant. All right, that's it. Again, we're pulling out fewer all white marbles because there's fewer white marbles in the population. So at this point in time, with every generation, is it evolving? Yes or no? Yes, no, yes. It is. So every time we change the allele frequency, every time we take out some white marbles, that is the population evolving. So we've gone through, I think, generation 1, 2, and 3. I just have 4 and 5. I have to do this 2 more times. Complete the table, I hope. My survivors, they go back in. White marbles, still dead. Oh, and one important thing is I'm not looking when I'm pulling these out. I'm looking at the screen right here, so I'm seeing it when it comes out to you. I'm not looking in the cup. I'm not choosing. It's a random pull. Just want to make sure you are aware of that. All right, generation 4. Table 5.1. Mixed it out and hetero. Dominant. Hetero. Dominant. Hetero. Dominant. 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 Hetero. Dominant. Hetero. 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 Dominant. Hetero. 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 Dominant. 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 Hetero. And the last pair, hetero. I don't remember. I don't think anyone died that generation. So that's it. That was generation 4. Mix it up one more time. And you go. Hetero, dominant, hetero, 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 dominant, 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 hetero, hetero, dominant, hetero, dominant, dominant, dominant goes in there, dominant, 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 hetero, hetero, dominant, 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 hetero, dominant, dominant, hetero, dominant, and hetero. Again, nobody died in that generation. All right, so hopefully you were able to record those data. Every time I say hetero or dominant or recessive, that should have gone into one of your four or one of the three columns on table 12-1 for generation 1, 2, 3, 4, and 5. And you'll notice, if you look over those data, I don't have the exact numbers. I wasn't keeping track, but I'm going to see a bunch of copies of this, so you better be right. We noticed that the number of homozygote recessive decreased very, very rapidly. We had a bunch in the first generation, a few in the second generation, I think 2 in the third generation, and if I recall correctly, none in the fourth or fifth generation. So the question then is, how many generations does it take, or is it even possible to get rid of deleterious alleles when they are recessive within a population? And this population started off pretty small. Started off with 46 individuals, 48 individuals. 48 individuals, if we counted it properly. So with only 48 individuals, we weren't able to get rid of the deleterious recessive alleles within 5 generations. And in fact, you probably never will, because then as the frequency of the recessive allele decreases within the population, the chances of getting an individual who's homozygous recessive decreases. Now imagine if you dealt with a population like the human population at 7.5 billion people. What are the chances that the last 2 people on Earth, out of over 7 billion people, would come together and form a homozygote recessive offspring? Unlikely. It's not going to happen. So it's almost impossible, if not impossible, to get rid of a deleterious recessive allele within your population. All the marbles are back in. So now what I'm going to do is do table 12-2.
So where we left off in the video that you watched was that in the first generation of the marble experiment, we were looking at deleterious alleles. And deleterious alleles are alleles that are lethal. And so in that experiment, the black marbles always represented the dominant, and the white marbles was always the recessive. And so in the first round, the recessive allele was the one that was deleterious, meaning that was the one that died off. And so what that represents is that the 2 white marbles together would be the ones that would die off, right? Because you have to inherit 2 recessive alleles to have the recessive phenotype. So in round 1, the recessive alleles were deleterious. Now, what you saw in that video was that in round 1, in 5 generations that we looked at, the white marbles did not disappear from the population. And that's because if you have heterozygous individuals, right, heterozygous individuals will keep that recessive allele in the population. That trait will continue to still be in the population. And again, it's not likely that you're going to ever get rid of that allele completely, because as the frequency of that allele gets lower and lower and lower, the chance of 2 heterozygous individuals coming together and producing that recessive offspring to eliminate it from the gene pool is not very likely. And so that's why we see so many recessive diseases that can stay in the population, because we can have instances where carriers will pass those traits on, and therefore those traits can stay within the population. In round 2 of this experiment, the dominant allele is going to be the allele that is deleterious. This is the one that's going to die off. So if the dominant allele is deleterious, which ones are going to die off? Which marble pairs are going to die off? Well, homozygous dominant is going to die, right? So the black-black combination is going to die because they have 2 dominant alleles, as well as the heterozygous. The black and the white together is also going to die, right? Because having just one dominant allele is going to be deleterious. That phenotype is going to be seen. And so what we're going to look at is we're going to look at when we draw our marbles, we want to look at and see how many generations does it take to remove the dominant allele from our population. And so let's look at that experiment. Okay, so he's starting with his population again, and he's shaking them up to make the process random. And he's going to pull out marble pairs, and you're going to record this in table 12-2. And so you're going to have your cups, right? So these are going to be your cups that you have, and you're going to record the number of homozygous dominant. So we have homozygous recessive, there's one, recessive, heterozygous, recessive, recessive, hetero, hetero, homozygous dominant, hetero, 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 homorecessive, homozygous dominant, hetero, hetero, homozygous dominant, hetero, 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 homozygous recessive, homozygous dominant, heterozygous, homozygous dominant, heterozygous, homozygous dominant, homozygous dominant, heterozygous, heterozygous, homozygous recessive, heterozygous, heterozygous, homozygous dominant, homozygous recessive, homozygous dominant, homozygous recessive, homozygous recessive, heterozygous, heterozygous, homozygous dominant, homozygous dominant, heterozygous, 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 homozygous recessive. And so now when you think about this, which of these are going to die off? And the answer is that anything that has the dominant is going to die. So look at your new population. The only thing that survives is homozygous recessive, because if it has the dominant allele, it's not going to be able to survive. And therefore, it only takes one generation to remove that from the population. So what we saw in that experiment is that if you have a dominant deleterious allele, that would be lethal within one generation, right? Because if the dominant allele is the one that is lethal, even being heterozygous is going to die off. And the only thing that survived in round 2 was homozygous recessive. So it only took one generation for that dominant deleterious allele to be removed from our population. Whereas in the first round, when the recessive allele was the one that was deleterious, that one, right, after 5 rounds, we still had the recessive deleterious allele in the population because the heterozygous survived. Now, there is an exception to the dominant allele being removed in the first generation, and that is that in some cases, dominant deleterious alleles aren't necessarily lethal right away, meaning it doesn't mean that the babies can't be born. It could be a condition, a dominant allele can be a disease that comes on late onset. So an example of this would be a disease called Huntington's disease. Huntington's disease is a neurological disease. It's a degenerative neurological disease. Huntington's typically comes on in many patients in their 40s or 50s. And so you have to think about it that if somebody can survive to their 40s or 50s, they've likely already passed on that dominant deleterious allele prior to getting the disease. So what that means is that they still have offspring and they pass that trait on to their offspring, but it's deleterious later in life. They still die early, but maybe not as early or not being born at all, right? And so in that case, if the deleterious dominant allele has a late onset, what that means is that that allele can stay in the population. And so that's why Huntington's disease continues to be seen, because by the time people are diagnosed with the disease or even know that they have it, they've likely already had kids, and those kids have passed on that trait. Another example would be achondroplasia. Achondroplasia is a type of dwarfism. If you've ever seen that show, Little People, Big World, you've probably seen that you can have two parents who have dwarfism. Now, I'm not sure if they have the achondroplasia specifically, but you can see this example there. You have two parents who have dwarfism, and they have normal height children. The reason for that is that achondroplasia is a dominant trait. So those two people who have dwarfism, they both carry the recessive allele. And by chance, they can both pass on those recessive alleles to their children and have normal height kids. Now, typically achondroplasia, people who are born with that, have a shorter lifespan than the general public, right, than most people. But again, not so deleterious that it kills them before reproductive age. And so in that case, they can pass that trait on to their offspring, and that dominant allele stays within the population. And so there are cases where a dominant deleterious allele can remain in a population for more than one generation. But again, it has to be something that comes on later in life and isn't deleterious until after reproductive ages. Because again, if they can reproduce, then they can still pass on that trait to their offspring. And so there are exceptions, but again, in this experiment, we were looking at that they would die right away. And so in that case, our dominant deleterious allele was wiped out of our population after one generation, because none of them that had the dominant allele would survive and pass that trait on to the next generation. And so that's what we were looking at in that experiment. Now, the next thing that I want to go over with you and spend a little bit of time on is talking about the graphs for the natural selection experiment for the predator-prey interactions. And so I just want to kind of go over with you, what does that graph look like so that you can have a general understanding of what this should look like.
Hi everybody, and welcome back. We are ready for part 2 of this lab. And in this part, what we're going to do is we're going to look at the effect of variation in mouth parts in both predators and prey. And we're going to track how that variation can influence how populations change over time. So I'm going to go ahead and pull up that screen and explain what's actually going on with this one. And so in this next part of the lab, what we're going to be doing is we're looking at how individual variation among a predator population can influence foraging success. And then we're also going to look at how variation within a prey population or within prey species can influence their survival in different types of backgrounds. And ultimately, again, this is a lab that is best done on campus with a large group of people. Up to 32 people is ideal for this one. And so what I would normally do is take the class and divide them into 5 separate teams, if you will. And each team represents a different type of mouth part within that group. And so some of the things that we typically use are rulers. We can use spoons, screwdrivers, tweezers or forceps, pliers, Sometimes we use chopsticks, sometimes we use forks. So there's a whole variety of different mouth parts that we can choose from. And ultimately then what we do is we go outside and I release the prey into the wilds of Orange Coast College campus. Typically what that means is I release them in the grass on the lawn, and then I give my students about 1 minute to forage for those mouth parts, for those prey items. And the prey items that we use for this lab are beans. And we have 5 different types of beans that we use. We use kidney beans, and I'll go ahead and pull those up real quick. Typically use our kidney beans, which are large, brown, red beans. We call them the red beans because they are kind of a reddish color. We use pinto beans, which are brown with little black spots on them. We use black beans, which, hey, they're black in color. We use, I don't even know what those are called. We call them the white beans. I think they're called northern something or other. I don't even know what they're called. They're the white beans. And then we also use split peas for our green beans. You'll notice that they're different in size. The red beans are the largest. The green beans are the smallest. They get progressively smaller in the order that I presented them. And what I did is I counted out 50 of each bean and put them into this little Tupperware container. In lab, we would normally use 150 of each bean. So we would actually throw 750 beans out into the grass. I have 50 of each, so that's 250 beans that I'm going to throw out. As for the various mouth parts for our predators, we can use chopsticks. We have screwdrivers. We have forceps or tweezers. I grabbed a pair of pliers, and also I grabbed a plastic spoon. So what I'm going to do is I'm going to go outside and try this. I'm not sure how it's going to work, but I'm going to go outside, I'm going to try this, and I'm going to spread all 250 beans out in the grass in my front yard or backyard, depending on where I can find some grass that looks kind of shitty so that it's not as lush and green and all the beans just disappear. I'm going to use my plastic cup that I had earlier for the marble example as my stomach, and then I'm going to actually film myself picking up beans with each one of these 5 different mouth parts, or 6, however many I have, 1 minute each. I'm going to count how many beans I capture, and I'm going to use that to essentially demonstrate that some are easier to use and more efficient than others. Ultimately, after we're foraging, the mouth part or the tool that's used for foraging that catches the most beans, well, they got the most food. So they're more likely to survive and reproduce. And so in theory, that population would grow over time. The mouth part that catches the fewest number of beans, well, they're least successful in foraging. They're going to have the least amount of food. So some of those individuals would probably die off. They're not going to do as well. So they might die off. And so again, if we were in lab, I would shift after each foraging round. I would shift the number of individuals within the class so that the most successful group, they actually get more individuals, so more people join that group. And the least successful group, some of those die off. So the people who die go to the successful group. And those populations then will shift or change from one generation to the next. Same thing happens for our beans, actually. Every bean that gets captured during the lab experience, they would actually die. So they would be removed from the population because they were killed and eaten. Any surviving bean, which means any bean that's left in the grass or on the ground after that first minute, well, those survive, and for each survivor, we're going to add 1 more bean to that. So those populations would potentially increase. So I'm going to go outside. I'm going to do this for a few minutes. My numbers are probably going to be low because I'm working solo, but I'm going to go ahead and pause this and get set up outside. All right, gang, I'm in my backyard right now. I've got all of my tools handy. I've got my 250 beans, and I'm just going to go ahead and spread those beans out in the grass. And I've got my phone started. It's got a 1 minute countdown on it. So what I'm going to do is I'm going to start that countdown, and I'm going to try to catch as many beans as I can of any type. Doesn't really matter which. And I'm going to start with the tweezers. And I'm going to see how I do on that one. So go ahead and start. Oh, that's time. All right, let me count what I've got here. So in terms of my beans, you don't have to record this on your data table, but I just want to demonstrate what I have. I just dropped a white bean. I'll pick that one up again. So I had, in terms of the kidney beans, 1, 2, 3, 4, 5, 6, 7, 8, 10 kidney beans is what I caught on that one, or 10 of the red. I have 4 white beans, 1, 2, 3, 4. I have 5 pinto beans, 1, 2, 3, 4, 5. 2 black beans, and 1 green bean. So that's what I caught on my first round. Mostly the pinto beans. Got 10 of those, 5 of the pintos, 2 black, 4 white, and 1 green. I'm going to go ahead and release these again, and then I'm going to try a second round with chopsticks. Don't worry, I won't let you let the beans go back out into the population. Cup is empty. Let me go ahead and pause this, and I'll let you know when I'm done. I just finished chopsticks. Here's what I've got. Pretty much all pinto bean, all red except for 2 pinto. So I've got 2, 4, 6, 8, 10 kidney beans again on that one, and 2 pinto beans. I didn't get any black, I didn't get any white, and I didn't get any green. All right, I'm going to go with this. All right, I've got my spoons, and I got a lot more that time. So let's go ahead and sort them out based on color. So again, I've got quite a few more beans on that one. I've got 1, 2, 3, 4, 5, 6, 7, 8, 14, 15, 16 red beans. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 pintos. 1, 2, 3, 4, 5, 6, 7 white beans. 5 black beans, and 3 green beans on there. So I got a lot more with the spoon than I did with the forceps or the chopsticks. I'm going to run through this one more time with the pair of pliers. I'm going to go ahead and release everything one more time. They're all released again. Pause. And I just finished with my pliers. Here's what I've got. A lot more grass in there. I wasn't as able to pick up as cleanly. So let's get rid of the chaff and see what we have for numbers. Got 13 of the red beans, so 13 kidneys, 7 pintos, 6 white, 0 black, and 1 green on there. So you'll notice that even though I started with the same population size, every time I started this, I had 250 beans to choose from, but by using the different mouth parts, they were different in their foraging success, or at least in my ability to use them. Let me go ahead and show you. I'll kind of just hold this over the grass for a minute. So it's fairly obvious. The kidney beans, they're large. They hang out, they're fairly obvious up on top. The black beans are significantly smaller. They tend to blend in a little bit more. The green beans, see if I can find one. There's 50 of them down here. There should be a bunch. Anyway, there's some pintos, there's some blacks. Not seeing any, oh, there's one. So the green beans, again, they're pretty much the same color of the grass, and they're small, and they blend in pretty well. I sure hope my chickens like to eat beans, otherwise I'm going to have a really interesting growth in my lawn in a few weeks. All right, so that's it. Let me go ahead and pause this one more time. I'll go back out to the campo, and we'll wrap this up. Okay, everybody, welcome back. I hope that was helpful in terms of getting an understanding of how the lab is typically run and what the challenges are with respect to using the different mouth parts, the chopsticks, the pliers, the tweezers, the rulers. And so what I'm going to do is I'm going to go ahead and jump back to the PowerPoint and then jump back to our lab report, page 174, and we're going to fill out tables 12-2 and 12-3. Now, the numbers that I'm going to present to you are actually numbers from a previous semester that I have on file. So we weren't able to go through and do all of this together, which is unfortunate because, again, it's actually quite a fun lab to do. But let me go ahead and pull up the PowerPoint one more time. With this PowerPoint, again, we were on this slide where we were getting ready to go outside and run the lab, and we had different mouth parts. Again, we consider this all part of the same species, but different alleles 4 different mouth parts, so we had variation. Obviously, these are extreme examples, but it works well to demonstrate the point that some mouth parts are more efficient than others. And what we would typically talk about before going outside is some of the rules of this game. Again, this didn't apply because I was playing by myself when I did this, but generally we don't want to hurt each other. So no biting, no kicking, no scratching of your classmates. That aside, competition is generally part of the game and par for the course. So when you've got a bean that you're trying to get into your cup and somebody else sees that, they're willing to, or they're welcome to try to steal that bean from you. So that happens particularly when the populations are getting lower or if they can't find their own beans. Some students tend to develop a little territory where they essentially block an area with their arms. There's a lot of variation that we see going on with that one. So I'm going to actually go back to open up our lab manual, and we're going to be again on page 174. And on page 174, what I want to do is actually fill out table 12-2 and 12-3. And again, I'm using numbers from a previous semester for this. And in this class for the previous semester, we had a total of 30 students. So I divided them equally into 5 groups, and I had 6 of each. So 6 forceps, 6 pliers, 6 rulers, 6 spoons. We didn't use screwdrivers that semester, but we used chopsticks instead, and we had 6. And based on the total number of kills, the groups that were more successful at killing or harvesting more beans, those were the groups that increased in number. And the groups that were less successful, they actually decreased in size. So that was our initial numbers for our parent generation, our first generation. After that first round, I'll give you the numbers. After that first round, I'll give you the numbers. I just paused. So here we go. The initial numbers were, I'll go ahead and just work it across, 6, 6, and 5. And then for the pliers, they actually did well. So 6, 7, 8, and they got up to 10 on there. For the rulers, they didn't do as well. 5, 3, and they dropped back down to 1. The spoons, they did great this particular semester. They did, so that's an 8, 6, 8, 11, and 14. And the chopsticks, they didn't really do as well as they hoped. They lost 2 every generation, and they actually went extinct. By the 4th generation, the chopsticks went extinct, and the rulers were very close to extinction. In fact, one individual, yeah, pretty much they were close to extinction. Now, if we did this in lab, again, our initial numbers for our first generation would have been 150 all the way across. In terms of the total number of kills, for green, it was 11. Red was 118. For black, they had 67, 86. For brown and white was 92. So in order to get the number of survivors, we'll subtract the total kills from our initial numbers, and that's 139, 32, 83, 64, and 58. Now to get the initial numbers from our second generation, we're just going to double our number of survivors or multiply that by 2. So our initial numbers, and what that means, if we were doing this in the field, whoever had the green beans as their group, they would have to count out 139 additional green beans so we can add those to the population. Red would count out 52, black 83, brown 64, and white 58. So our initial numbers, we're going to double everything. So 278, 64, 166, 128, and 116. For our total kills, 23, 51, 72, 53, and 70. Our number of survivors, again, we're just subtracting. So that was 255, 13, 94, 75, and 46. And then our initial numbers, doubling everything again, 510, 26, 188, 150, and 92. Total kills for that third generation, the third time, is 38, 26, 91, 68, and 49. And the number of survivors, 472, 0, 97, 82, and 43. If we did this a fourth generation, the initial numbers, if we went through this one more time, we would start with 944 green beans, no red because they were extinct, 194 black, 164 brown, and 86 white. So those are our numbers for us.
There we go. So this is the data table for your experiment. And so this is your table 12-3. And what you're looking at is the predator populations over time. And so when you do your graph, you're going to have two axes, right? You're going to have your population size, right? Your population size. And you're going to have, so your population size and by generation, meaning we're going to look at how the number of predators changed over generations. So if you think about this, right, if we're thinking about a graph and our X and our Y, we want to think about which one is our dependent and which one is our independent. So does the number of predators, so the predator population, does it depend on the generation, or does the generation depend on the number of predators? Well, the generation time can't change depending on the number of predators. So it's predators, the number of predators, that depends on the generation. So your predators are going to be your dependent variable. Those are going to be on your y-axis, and you'll see this in a minute. And you're going to have your generation, which is like time, on your x-axis, because that is your independent. So what you're looking at is you want to look and see what happens to these populations over time. So if I look at the forceps, right, in the first generation or the parental generation, we started with 6 forceps. After one generation, we still had 6. In the third generation, we had 6. And in the fourth, we had 5. So our population decreased a little bit. In our pliers, it was 6, 7, 8, and then 10. So the pliers were somewhat well adapted because their population was increasing over time. So what you want to think about is what type of graph would best represent this, right? What type of graph should we do for this type of data? If we're measuring something over time and the data is continuous, what type of graph would we use? The answer is going to be a line graph, right? We want to look at these populations over time, and therefore that would be continuous data, and we should use a line graph. Now the next thing, how many lines are you going to have? Well, we looked at 5 different predator mouth parts. We looked at forceps, pliers, rulers, spoons, and chopsticks. So how many lines are you going to have on your graph? And the answer is you are going to have 5 lines. 5 lines. So we'll talk about that more in a minute. Now let's look at the prey table. Here is our prey table, right? And so what we're looking at is the prey population over time. So when you're graphing this part, it's a little bit confusing when you look at this to know which part you should be graphing. Again, we're looking at the prey population over time. So when you're graphing it, you're going to graph these initial numbers. So when we start, the green was at 150. In the second generation, we're going to plot 278. In the third generation, you're going to plot 510. And in the fourth generation, you're going to plot 944. So when you're looking at this table, I know it looks really busy and it's hard to figure out what to graph, but you're going to graph the initial numbers each time. So your parental generation, your second generation, use the initial numbers, then the third generation, your initials, and the fourth. Because you'll notice if you look and you compare, depending on the number of survivors, right, each of those survivors that survived, they reproduced, and that gave us the next initial number. So in the first generation, right, the number of survivors for the green beans, there were 139. So if each of those represented or reproduced, the next population size would be 278. So in the second generation, there would be 278 individuals. And then in the third generation, there would be 510. And in the fourth generation, there would be 944. So those are the numbers that you want to graph. You want to graph in this table the initial numbers every time. You don't want to graph all the rest of it. So it's just the initial numbers that are going to be graphed. So here are our graphs that we're going to look at. So again, we're going to have two graphs for this. We're going to have one that's looking at the predator population, and we're going to have one that's looking at the prey population size. So I gave you some terms that you might want to include in your title. So predator, right? So if you're talking about the predator population, so predator, you want to have mouth part, right? Because you're looking at the effect of various mouth parts, and you should have natural selection in your title. In your second graph, the terms that you want to include prey, right? Because you were looking at the prey population. You want to include the term beans because we were looking at the different colored beans. And then again, you want to include the term natural selection. So make sure to incorporate those terms into your title in some way. So let's start with the predator population. So again, we said that the number of predators, so our predator population size, depends on the generation. And so our predator population size is going to go on our Y, and our X is going to be, and the X is going to be the generations. And so that's what we have here. We have our Y-axis has our predator population size, and our X-axis has our generation. Now, if you look at your table, right, if you look at that table, you need to look at what is the largest number that you had for your predator population size. And the largest number that you had in your table was 14. So when you make your scale, you want to make sure to make it so that it can fit on the paper. You want it to take up most of the paper. You don't want it to just occupy one little corner, most of your graphing paper. And so use your scale accordingly. And so what I did was I put on the Y or the X-axis, we have the parental generation, we have the second, third, and the fourth. And then my scale for my predator population on my graph paper would be 3, 6, 9, 12, and 15, because again, 14 is my highest number. So I would set my graph up like this. Now again, we said that this needs to be a line graph, right? And how many lines are we going to have? Remember, we're going to have 5 lines. So if I were to take and look at the first data, so what I had was for my first mouth part, the numbers were 6, 6, 6, and 5. And so those are my data points. And so I would put my data points on and then connect my dots. Now you can do this with different colors. So you could represent them. You don't need triangles. You could do like just dots and make them different colors. That's one way to differentiate your lines. Another way is to use different shapes. So let's say you didn't have colored pencils at your house. Well, don't worry. You can differentiate your lines not by color, but instead by shape. And so let's look at the next data point. So let's say we look at the next mouth part, right? It goes 6, 7, 8, and then 10. So I would then connect my dots. Again, mine are color coded. You would use straight lines and a ruler, okay? But this is the general idea. And so you can, again, you can color code them. You could just use dots and use different colors. You could use different shapes. So like those could be in black. They could all be in pen, black pen, as long as I use different shapes for the different data points. So you have to be able to differentiate your lines in some way. So on this graph, I'm only going to put 2 of them, but know that when you're done, you should have 5 lines. Now, I have this. What is missing from my graph still, right? I have my terms for my title, so I'm not including the title because you're going to write your title. But what else do you need to have in your graph to make your graph complete? And the answer is you need a figure legend. You need a key that tells somebody what do the different lines represent. So my triangles were my forceps mouth part. My pliers were the rectangle, and so on and so forth. And so you need to make sure that you have a figure legend that basically tells somebody what are the different lines, which mouth part does that represent. And so you're going to end up with 5 different lines on your graph. And this is going to allow you to see which mouth part was best adapted to their environment, right? That would be the one where the population increases, whereas the one where the population decreases would be the least adapted. And that's what we're looking for. Now, in our prey experiment, the graph is set up in very much the same way. We're comparing prey population size, right? So our prey population size and generation. So my axes are still the same. So I have, oops, I didn't change that. That should say not predator, that should say prey. Sorry about that. That should say prey population size. So my prey population size would be here. Generations would be on the bottom. And then again, I need to look at my prey population sizes. And you'll notice that you had one that was in the 900s. So you have to make your scale accordingly. And so if I was doing this, I might make this scale 200, 400, 600, 800, 1,000. And then my generations would still be parent, second, third, fourth. When I do this, again, I have 5 different types of preys. I have my different bean colors. I have my green, my red, my white, my brown, and my black. So how many lines should you have on this graph? Well, you should have 5 lines, right? 1 line for each of the different bean colors. And again, just like the predator graph, you want to make sure that you differentiate your 2 lines and also include a figure legend that tells somebody what the different lines represent. And so this is what you need to do for your graph. Include those terms on your title and then set up your graphs and make your graphs for your 2 parts. You need one for the predator population and you need one for the prey population. So that's what you're doing for your graphs.
So with the PowerPoint, with where we left off, we had our mouth parts, we went through the rules, and I just took 2 different pictures of beans. These are actually the same number of beans. I didn't count out 250 each time, but I made sure I had the same number of each color, and I threw these down in different types of habitats, just to give you a visual of what each one might look like. So taking a look at this picture, what are the colors that stand out the most, or the beans which are the most obvious? So obviously the kidney beans, those are really obvious in this case. Some of the pinto beans, this one right here was a pinto. The pinto beans aren't as obvious as the kidney beans are. The green beans actually here are fairly obvious. They're actually quite easy to see. So unlike in the grass where the green beans pretty much disappeared right away, here they're actually relatively visible. You see the white beans are standing out quite a bit as well, but you'll notice in this case that the black beans are hiding. They're doing a very good job of hiding in this particular color combination, in this color scheme. The soil is darker, and when the soil is darker, we can expect there to be a different selective pressure or selecting for a different color. The green beans did really, really well on the grass because the grass was green. The green beans were small. They could blend in both with color and their size made them fall through. And yet the kidney beans, the red beans, they didn't do very well there at all. If we had done the experiment on dirt like this, the green beans wouldn't have that same advantage. Even though they're small, the color contrasts significantly. So the black beans would have actually been selected for in this case. I took another picture. This is just a little bit further away, but the same general area. There was more leaf litter in this area, and you can see again the variation that we would see here. The beans which are, again, obvious, we've got kidney beans. Those still are pretty easy to find. The white beans, those are fairly easy to find. They stand out quite a bit as well. The pinto beans, where are they? Oh, there's one. There's one of the pintos. The black beans are a little bit easier to see now here than they were earlier, but the pinto beans are really, really difficult. Turns out, if you look right here, I've got a pinto bean, and it's almost the exact same color as the pine needles that are lying on the ground as well. So in different types of habitats or with different backgrounds, different colored beans would be selective. So think about that with respect to camouflage and prey. If these were, instead of just being beans, but if the beans were animals of some sort and they were relying on that camouflage to avoid being eaten, depending on which habitat they were in, sometimes they would do significantly better in certain habitats and then potentially worse in other habitats. The white beans were pretty much obvious all the time, but imagine if this were done in winter and there was snow on the ground, or on an area where instead of being brown soil, maybe it was sandy soil, which has a very light color. In that case, the white beans might actually blend in quite a bit better, and the other colors would stand out more. So the background of the environment has a significant impact on the ability for these beans to blend in and their camouflage.
So what I put here is your Quiz 12 study guide. So for natural selection, you want to define natural selection. You want to understand that a trait would be passed on and selected for if an individual with that trait has more offspring than other individuals. So when you think of natural selection, it's not simply just survival of the fittest. That's not exactly entirely correct, because it's not only survival, but it's differential reproductive success. Organisms that have more offspring are going to pass those traits on with greater frequency than an organism that has less number of offspring. And so again, it's not just survival that matters. Yes, survival definitely does play a role, but it's not limited to only survival. It has to also include reproduction, right? And it has to be a trait that is heritable, because if that trait can't be passed on, but it gave somebody an advantage, well, you're not going to see that trait more likely in future generations because it's not heritable. So it also has to do with the trait being heritable. Understand that sexual reproduction provides genetic diversity, which can affect natural selection, right? Because when you have natural selection, you get unique combinations of genetic possibilities. And when you have genetic diversity, right, that can affect natural selection. In your marble experiment, if you're given pairs of marbles and told the pattern of inheritance of deleterious alleles, meaning told whether it was dominant deleterious or recessive deleterious, you should know and understand how many survivors would be left, right? So if it was a recessive deleterious allele, then only the whites would be killed off. If you're talking about a dominant deleterious alleles, homozygous dominant and heterozygous would both die. So if I gave you, a certain number of pairs of marbles, you should be able to tell me, if I told you its pattern of inheritance, how many survivors would be left. How is it possible for a dominant deleterious allele to remain in a population after one generation? Again, I talked about this, that if that dominant deleterious allele is something that comes on later in life, well, then those people who have that dominant deleterious allele can reproduce and pass it on to future generations. So it is possible for a dominant deleterious allele to stay in a population after one generation, but in our experiment, we only saw it in one because we assumed that they died off right away. Is it likely for a deleterious allele to be removed from a population? And we said that no, it's not very likely, right? Because in order for it to be removed completely, one, heterozygous individuals are going to keep passing on that trait and it's going to stay in the population. And then for the next part, for recessive deleterious alleles, what happens to the frequency of that allele in the population? Well, as those homozygous recessives die off, right, the frequency of that allele in the population gets low. And when the frequency gets low, it's less and less likely that two individuals who are heterozygous are going to come together and have a child, which would remove that gene from the population. So it's not very likely that a deleterious recessive allele is going to be removed from a population. For the predator-prey experiment, you want to review what behavioral adaptations helped predators to get food. And so that was covered in the video, right? So territorial, aggression, etc. So you want to review what some behavioral adaptations were that helped predators to get food. What represented the prey in this experiment, right? The prey in our experiment were the different colored beans. So those represented our prey. In our predators, we were looking at the effect of the mouth parts on their ability to get food. And then lastly, if given a graph, know which predator preys are best or worst adapted. Meaning, if you see the population size going up, that means that they're well adapted. If you see their population size decreasing, that means that they're not well adapted and their population size is decreasing. And at some point, that population could potentially become extinct, which you want to see, did we see that in our experiment? And then lastly, read Lab 13 that covers anatomy and physiology. And so this concludes the summary for Lab 12.